Welcome back to Clinical Hematology Reviews. Today we'll be discussing erythrocytes. Red blood cells are far more than passive bags of hemoglobin. They're exquisitely engineered specialized cells optimized for oxygen and carbon dioxide transport throughout the systemic circulation.
That is so true. At its core, the erythrocyte's primary physiological job is taking up oxygen in the lungs, binding it securely to hemoglobin, and delivering it to systemic capillary beds to feed tissue metabolism. On the return trip, red cells assist in gathering carbon dioxide from metabolizing tissues and transporting it back to the pulmonary circulation for elimination. When this cellular balance is disrupted, we see clear clinical manifestations. Anemia, for instance, represents a drop in red cell mass or functional hemoglobin, directly producing tissue hypoxia. On the flip side, erythrocytosis is an absolute or relative elevation in circulating red cell concentration.
And clinically, distinguishing relative from absolute erythrocytosis is vital. Relative erythrocytosis occurs when plasma volume drops, such as in severe dehydration, causing a hemoconcentration that makes the red cell count appear high without an actual expansion of total red cell mass. Absolute erythrocytosis, however, stems from genuine cellular overproduction. A classic primary example is polycythemia vera, driven by the point mutation known as JAK2V617F. This somatic mutation causes constitutive activation of the erythropoietin signaling pathway, driving uncontrolled red cell proliferation in the bone marrow regardless of actual tissue oxygenation needs.
Looking at the complete lifespan of these cells, erythropoiesis begins in the active red bone marrow under tight systemic control. The primary hormonal driver is erythropoietin, or EPO, a glycoprotein produced mainly by interstitial cells in the kidneys. When renal tissue detects hypoxia, EPO secretion surges, stimulating red cell progenitors to proliferate and mature. Once fully mature, erythrocytes exit the marrow and circulate in peripheral blood for an average lifespan of 100 to 120 days, which is roughly 3 to 4 months.
Throughout those 4 months, the red cell travels hundreds of miles through turbulent arterial flow and narrow capillaries without a nucleus or protein synthesis machinery to repair ongoing damage. As the cell ages, its metabolic enzymes deplete, its membrane loses flexibility, and it becomes senescent. These aged cells are continuously monitored and removed by reticuloendothelial system macrophages. The primary filtration organ for senescent red cell removal is the spleen, specifically within the cords and sinuses of the red pulp. Secondary sites include the liver and bone marrow. Interestingly, if a patient undergoes a splenectomy or suffers from severe splenic atrophy, the liver's resident Kupffer cells step up to take over the main burden of culling old erythrocytes.
As precursor cells progress through erythropoiesis in the bone marrow, they follow extremely consistent morphological rules that help us identify them under light microscopy. Across the maturation spectrum, overall cell size decreases, the nuclear to cytoplasmic ratio drops dramatically, and nuclear chromatin condenses into a dense clumped chromatin, eventually becoming a solid, structureless pyknotic nucleus before extrusion. Cytoplasmically, immature cells start out intensely blue due to high ribosome density, but gradually transform into a salmon pink color as hemoglobin accumulates.
Exactly. The sequence begins with the pronormoblast, also termed the rubriblast. This is a large cell with a very high nuclear to cytoplasmic ratio, deeply basophilic blue cytoplasm, a pale perinuclear Golgi zone, fine lacy chromatin, and 1 to 3 prominent nucleoli. Next comes the basophilic normoblast, or prorubrocyte. Here, the nuclear to cytoplasmic ratio is moderately high. The cytoplasm remains intensely blue due to abundant ribosomes, and chromatin condenses into a distinct spoke wheel arrangement, while nucleoli disappear. A pale perinuclear halo, marking the location of active mitochondria, is often visible.
Following that is the polychromatic normoblast, or rubrocyte. This stage marks a major transition point. The cytoplasm displays a gray-blue hue as significant hemoglobin production begins to blend with residual blue RNA. The chromatin becomes heavily clumped, and most importantly, this is the final stage capable of completing mitosis. In healthy bone marrow, polychromatic normoblasts represent the most abundant erythroid precursor stage. The final nucleated stage is the orthochromatic normoblast, or metaerythrocyte. Its cytoplasm is predominantly salmon pink due to high hemoglobin concentration, and its nucleus is fully condensed, dark, pycnotic, and displaced eccentrically, preparing for imminent expulsion from the cell.
Once the pycnotic nucleus is extruded, the cell enters the reticulocyte stage. Reticulocytes are anucleate. They retain residual organelle remnants, specifically ribosomal rRNA and mitochondria, for about 1 to 2 days in the marrow and another day in peripheral blood while completing final hemoglobin assembly. On a standard right gene-sustained blood smear, young reticulocytes appear slightly larger than mature cells, with a distinctive gray-blue tinge, a feature we record qualitatively as polychromasia.
However, to obtain an accurate quantitative reticulocyte count for assessing bone marrow erythroid activity, we cannot rely on standard blood stains alone. We must perform supravital staining using non-fixed, living red blood cells. Supravital dyes, such as new methylene blue or brilliant crescel blue, penetrate the unfixed cell membrane and cause residual ribosomal RNA to cross-link and precipitate into visible, dark blue reticular networks or cytoplasmic inclusions. Counting these stained cells allows us to precisely evaluate effective erythropoiesis in response to anemia.
Once the reticulocyte clears its remaining organelles, it becomes a fully mature erythrocyte, or discocyte. Anatomically, it's a flexible, biconcave disc measuring about 7 to 8 micrometers in diameter, with a central area of pallor occupying approximately 1/3 of the cell's center. Because it lacks nuclei, mitochondria, and endoplasmic reticulum, the mature cell cannot synthesize new lipids or proteins. Its structural survival depends entirely on the integrity of its membrane architecture.
The red cell membrane is a highly organized phospholipid bilayer protein complex that is roughly 52% protein, 40% lipid, and 8% carbohydrate. This complex structure serves 3 essential functions: maintaining osmotic equilibrium, facilitating passive gas exchange while controlling ion flux, and providing the mechanical strength and extreme flexibility needed to withstand high shear stress in the arterial system and squeeze through tight capillaries and endothelial slits in the splenic cords.
The protein component is divided structurally into integral proteins that traverse the lipid bilayer and peripheral proteins that line the inner cytoplasmic surface. Transmembrane integral proteins carry extracellular carbohydrate chains rich in sialic acid, giving the erythrocyte a strong net negative surface charge known as the zeta potential. This negative charge causes circulating red blood cells to electrostatically repel one another, preventing spontaneous cellular aggregation in blood vessels.
Band 3 is one of the most vital integral proteins. It acts as an anion exchange channel, facilitating the critical one-to-one transport of chloride and bicarbonate ions across the membrane, which is central to systemic carbon dioxide transport. On the cytoplasmic side, peripheral proteins assemble into a dynamic meshwork cytoskeleton. The principal structural protein here is spectrin, composed of alpha and beta chains coiled into a flexible, spring-like helical dimer. Spectrin dimers line up end-to-end to form tetramers that run parallel to the membrane. Spectrin is securely anchored to the band 3 integral protein via ankyrin, creating a resilient internal scaffold that allows the cell to deform and immediately spring back into its resting biconcave shape.
Looking at the membrane lipids, approximately 95% of total membrane lipid content consists of an equal 1-to-1 molar ratio of unesterified cholesterol and phospholipids. Because mature red cells cannot synthesize new lipids, they rely on passive, continuous lipid exchange with circulating plasma lipids, a process regulated by liver metabolism. When systemic lipid metabolism is altered or membrane lipid proportions shift, characteristic red cell morphological variants emerge.
That lipid exchange dynamic explains several distinct placical poikilocytes. For instance, large target cells, or macrocodocytes, occur when there is a proportional increase in both cholesterol and phospholipids, expanding total surface area relative to cell volume. Acanthocytes, on the other hand, arise from an increased cholesterol to phospholipid ratio, leading to excess cholesterol accumulation in the outer leaflet that forms irregular, asymmetrical, spiky projections, a common finding in severe liver disease. Echinocytes, or Burr cells, result from outer lipid leaflet expansion relative to the inner leaflet, producing short, uniform, regularly spaced blunt projections. Conversely, inner leaflet expansion causes stomacytes, characterized by a slit-like central pallor resembling a mouth. Furthermore, intracellular calcium plays a huge structural role. Approximately 80% of intracellular calcium is bound to the inner membrane. If intracellular free calcium rises, it induces skeletal protein cross-linking, markedly reducing deformability and causing premature splenic destruction.
The erythrocyte membrane exhibits precise selective permeability. It is freely permeable to water and anions like chloride and bicarbonate, but almost completely impermeable to mono and divalent cations such as sodium, potassium, calcium, and magnesium. Maintaining steep cation gradients between the cytoplasm and blood plasma is vital for preventing osmotic lysis and preserving normal cellular volume.
For example, potassium is concentrated heavily inside the cell at approximately 100 millimoles per liter, compared to only about 4 millimoles per liter in plasma. Sodium is kept low intracellularly and high in plasma. This difference has major clinical implications. In vitro hemolysis releases high concentrations of intracellular potassium into the specimen's serum or plasma, causing falsely elevated clinical chemistry results. To maintain these gradients, the cell utilizes active transport pumps powered by ATP. The sodium-potassium ATPase pump cleaves one molecule of ATP to extrude 3 sodium ions from the cytoplasm in exchange for bringing 2 potassium ions into the cell. Meanwhile, glucose enters the cell independently of ATP through facilitated diffusion via GLUT1 transporters down its concentration gradient. However, in severe clinical hyperglycemia, high intracellular glucose can draw excess water into the cell during automated instrument dilution steps, producing transient swelling that falsely elevates the reported mean corpuscular volume, or MCV.
Because mature red cells lack mitochondria, they depend entirely on specialized anaerobic pathways for glucose metabolism. Approximately 90 to 95% of cellular glucose is processed through the Embden-Meyerhof pathway via anaerobic glycolysis. This pathway yields net ATP, which fuels the active cation pumps, maintains membrane lipid structure, and preserves cellular deformability.
The remaining 5 to 10% of glucose is funneled through 3 vital metabolic shunts. First, the hexose monophosphate shunt, or HMP shunt, utilizes the enzyme glucose 6-phosphate dehydrogenase, or G6PD, to generate NADPH. NADPH keeps cellular glutathione in a reduced state, neutralizing reactive oxygen species. Without sufficient G6PD activity, oxidative stress causes hemoglobin to denature and precipitate into rigid Heinz bodies. Second, the methemoglobin reductase pathway uses NADH generated during glycolysis to maintain hemoglobin iron in the functional ferrous state, Fe2+, preventing the buildup of non-functional ferric hemoglobin, Fe3+. Third, the Rappaport-Lubering shunt diverts glycolytic intermediates to produce 2,3-bisphosphoglycerate, or 2,3-BPG. 2,3-BPG binds directly to the central cavity of the hemoglobin tetramer, stabilizing the deoxygenated structure and lowering oxygen affinity so oxygen is readily released into hypoxic peripheral tissues.
Finally, at the end of the erythrocyte's 120-day lifespan, cellular destruction occurs through 2 pathways, extravascular hemolysis and intravascular hemolysis. Extravascular hemolysis accounts for approximately 90% of physiological destruction. Aging red cells lose membrane elasticity and enzyme function, becoming trapped in the reticuloendothelial macrophages of the spleen, liver, and bone marrow.
Within the macrophage, the hemoglobin molecule is broken down and efficiently recycled. The globin chains are degraded into individual amino acids and returned to the systemic amino acid pool. Heme iron is liberated and stored locally as ferritin, or hemosiderin, or exported back into plasma bound to transferrin for transit to erythroid marrow. The remaining porphyrin ring is cleaved into carbon monoxide, which is exhaled by the lungs, and biliverdin, which is reduced to unconjugated bilirubin, bound to plasma albumin, and transported to the liver for conjugation and biliary excretion.
By contrast, intravascular hemolysis accounts for only about 10% of normal senescence, occurring directly within circulating blood vessels due to physical turbulence or mechanical microtrauma. When a cell lyses in the bloodstream, free hemoglobin dimers are immediately bound by the plasma scavenger protein haptoglobin, forming a large complex cleared rapidly by hepatic macrophages.
In cases of acute or severe intravascular hemolysis, plasma haptoglobin capacity is quickly overwhelmed, leaving haptoglobin levels severely depleted or undetectable in laboratory testing. Unbound excess heme then binds to hemopexin. If both scavenger systems are saturated, free filtered hemoglobin passes into the renal tubules, where tubular epithelial cells absorb and store the iron as hemosiderin. As these iron-laden tubular cells are shed into the urine, hemosideриnuria can be detected using Prussian blue staining on urine sediment, providing a classic marker of severe intravascular destruction. This brings our comprehensive review of red cell physiology, structure, and metabolism to a close. Thanks for joining us.